Thank you. I would like to begin by thanking the AACR for giving us this opportunity to highlight our research. In this slide, you will see at the top our title, the authors at the bottom, and then some background information which I will discuss. I'd also like to acknowledge that two of our contributing authors are here, Dr. Mark Pegram from University of Miami and Mr. Kevin Ellenson, U.S. Business Manager from Baumbach. The work we present today contributes to our biological understanding of well-documented disparities in breast cancer morbidity and mortality in several minority populations, particularly African Americans. For over a decade, our group has worked on further understanding the genetic basis of breast cancer in women of African ancestry. The studies we discuss today will focus on gene expression profiling in normal and cancerous breast tissue samples from a cohort of South Florida African American Hispanic and Hispanic women. Gene expression or molecular profiling has opened our eyes to the world of genetic and biological heterogeneity in many cancers. In breast cancer, it has led to subtyping of breast cancer into 5 major cohort types, which are shown here by receptor status such as ER, PR, and HER2/neu, and gene signatures. Basal-like breast cancer which is also referred to commonly as triple-negative breast cancer, is known as a particularly bad type of breast cancer. It is generally more aggressive and has no conventional therapy at this point. Strong evidence has accumulated over the years for an overrepresentation of basal breast cancer, triple-negative breast cancer, in African American women. Shown here are published results from the Carolina Breast Cancer Study that shows overrepresentation of basal breast cancer in both younger and overall African American women. This sets the stage for our study. So the questions we've addressed in this study are there ethnic-specific gene expression patterns among African American, Caucasian, and Hispanic women in breast cancer tissue and also in normal tissue? What are the pathways and molecular mechanisms behind the ethnic patterns or differences in gene expression? I will quickly walk you through this rather complicated slide, but our work starts with paraffin-embedded tissue, archived tissue. We've been able to partner with the company Almac Diagnostics, who has developed a specific DNA microarray, the breast cancer DSA, that is both enriched for sequences that are expressed in breast and also perfected for use with archived residual tissue. This has opened up a whole new world in terms of being able to use samples that were banked in the past for samples and bring them now to the contemporary world for genetic studies. For each sample, for tumor, we had tumor and adjacent normal for the tumor normal samples, and the normal samples I'll talk about in a minute. Typically, we get about 22,000 transcripts that come off of the hybridization analysis. We then run them through a very detailed data analysis, which is shown here on the bottom. And here we are comparing through a variety of statistical tests, comparisons of African American versus Caucasian, or Caucasian versus Hispanic, Hispanic versus African American, and getting significant genes that are 1.5 to 2-fold differentially expressed. We then put those in a path analysis and also cluster analysis. This slide summarizes the results of both studies. The normal study is brand new data I'm presenting here at this meeting. So you will see here that we are looking at now an average of about approximately 10 samples per group, African American, Hispanic, and Caucasian in the tumor here. And over here is the adjacent normal, again, averaged for the corresponding samples. This is what we see for typical gene expression profiling, where red indicates overexpressed genes and green indicates underexpressed. We have found distinct patterns that are different for each ethnic group in the tumor samples, African American, Hispanic, and Caucasian, and also some differences in the adjacent normal as well. And you can see that these, I'm showing you the various patterns here. On our presentation, which is on Wednesday morning, we'll show more of the data in that poster. I invite you to come there. I can tell you that in the African Americans, one of the samples that is overexpressed is in the DNA repair pathway, and we're very interested in that. Correspondingly, we are now looking at a number of normal reduction mammoplasty samples, and I'm presenting the pilot data here, from about 5 of African American, Caucasian, and Hispanic. Again, from that very large number of genes that we can potentially analyze, we only found a very small number that looked to be significantly different in normal breast tissue. And interestingly, some of these are as high as minus 10-fold difference. And so we're quite excited by these results. The next stage in this is we're going from normal FFPE samples to a new technique called laser capture microdissection, where we're going in and dissecting out the ducts and epithelial cells and the surrounding stroma microenvironment to look at potential expression differences in different parts of the breast. So then in summary, I want to emphasize these are preliminary data, which are being further investigated and validated with additional samples in a larger study cohort. It does appear that there may be ethnic-specific differences in gene expression in both normal and cancerous breast tissue, implying biological differences. The cause is yet unknown, but we are obviously continuing to work on this. We believe that we have demonstrated reproducible differences in gene expression between tumor and normal tissue from archived paraffin-embedded samples, and this is, of course, very important for going on for further studies. If the study results are confirmed, this offers new insights and hopes for ethnic disparities in breast cancer. And lastly, I have to say from a public health perspective, our study indicates the need and importance of accurately denoting ethnic status in medical records for future studies. Thank you.